Hello, everybody. In this lecture, we're going to talk about the replication of bacteriophages. So let's get right into it. By the end of this module, you should be able to explain the features of bacteriophage lytic and lysogenic replication and define the term phage conversion and discuss why it's medically important. And so viruses exist by hijacking host cell machinery. They don't generally have their own enzymes, although a couple package a few enzymes with them. And so what they do when they infect is they get into the host cell and take over their DNA replication material and their um, protein production machinery and get that to build new viruses. And so they rely on the host to replicate. In the lytic cycle, the basic way that this works is that the host cell is going to get infected by your bacteriophage. It's going to attach to the outside and insert its genome, immediately start building new virions. And then at the end, after those new virions have been completely built, it will lyse the cell to release the virions into the environment. And so we've got this 5-step process for lytic cycle, which goes from attachment to penetration to replication, assembly, and release. When we look at it a little bit more closely, the attachment occurs when the phage is going to bind to the outside of the cell. There's going to be specific receptors on the outside of the cell that the bacteriophage exploits. And then it's going to penetrate the cell by injecting its genome into the host. And then on the outside, you're going to have an empty, lifeless husk of a capsid just sitting there. So this picture on the right is showing that phage genome being inserted into the cell. And when we come over to the next stage, we've got the replication of that genome. So the genome is going to, at some point, find a ribosome, and that ribosome is going to be used to make proteins, or it's going to find, it's going to make its way all the way into to where the DNA polymerase and other replication machinery is located. And then you're going to just replicate those copies of the genome and make all the proteins that they code for. Now, the other kind of insidious part is that they're either going to be DNases, which are enzymes that break down DNA that are packaged with the genome, or they're going to be coded for on the bacterial genome. And those DNases are designed to break down the host genome. That way that they can be focused on for their own replication. Once everything is built, the genome will be packaged inside of the capsid and the remaining structures will be built. And then they are released back into the environment through a process of lysis. And those new phages will float around until they come into contact with another bacterial cell. And so here's a review of how that process works from attachment to release. Now, in the lysogenic cycle, you get what are known as temperate phages. And so they have a similar process where they attach and then penetrate. But then the phage genome is actually going to be incorporated into the host cell genome, forming what's called a prophage. And so a prophage is where the genome of the viral cell is getting replicated with the cell itself, but you're not getting the actual assembly of new virus. And then if the host cell is stressed, then you can get excision of that DNA, resulting in the packaging of new virions. And you go directly back into the lytic cycle. And so here again, you have attachment and penetration, just like we do during lytic replication. But here, the genome goes and gets integrated into your host genome. And that's represented by the yellow in this image. as the host genome or as the host replicates and you get daughter cells, each of those daughter cells are going to carry the host or is going to carry the viral genome with it. Then at some point, again, you hit that stressor, which is going to result in the DNA being excised, the phage DNA being excised, and ultimately going back into that lytic cycle where you replicate the genome, assemble the virions, and then lyse the cells. And here, this is the lysogenic, or this is the lytic cycle phase again. And next we have something called phage conversion. Prophages are interesting because there are actual organisms that have viral genome or viral DNA in them, including ourselves, that basically never was able to excise itself. Some of these can actually add additional properties to the host. And so phage conversion occurs in some pathogenic bacteria or possibly makes them pathogenic by giving them additional virulence factors. And two examples of this are the diphtheria toxin and the botulinum toxin from Corynebacterium diphtheriae and Clostridium botulinum. And so at this point, you should be able to explain the features of the lytic and lysogenic cycle and define phage conversion and discuss why that can be important. I hope you enjoyed, and I'll see you soon.